The Fluorescent Microscopy and Imaging Center is a centralized resource here at NIEHS that allows researchers within the Division of Intramural Research as well as the Division of Translational Toxicology to have access to cutting-edge microscopes. Predominantly, we focus on fluorescent microscopes, confocal microscopes, as well as some light sheet and super resolution microscopes. One of the unique benefits of being a core facility is not only do you have world-class equipment located together in one area, but then you also have personnel that are 100% dedicated to support the equipment and the training of the people using it.
The facilities there are second to none, and the support provided there by the staff is absolutely outstanding.
These researchers will come in on their own and operate the microscopes by themselves, and that is through the training that the staff of the Fluorescence Microscopy and Imaging Center have given them. When you come through the door and you need training, it doesn't matter whether you're an expert in using a microscope or you really don't know which end of the microscope to look into. We're going to go through how a fluorescent microscope works, how the optics of a confocal microscope works, how you can choose your dyes, and how you should set up your experiment and go through controls. Training for the hands-on part usually takes around 2 hours to be proficient, to be able to acquire an image. Over the course of months, as we learn more about an individual's project, then we will begin to suggest new methods or advanced approaches that they can use. And then we start digging into the part of image analysis.
I'm still learning how to use MRs, and that's why I feel like super comfortable working with you and with Erica, because every time that I have a question, it's like, oh my gosh, I'm struggling here. Like someone comes to the rescue.
The support that they've provided has been absolutely tremendous, and we couldn't do our research without them.
These microscopes are fairly complex. Price-wise, some of our microscopes cost around $100,000, and then they actually go up to about $750,000. Researchers know that they can send the staff to their individual lab here, and that when images are acquired, there's going to be great confidence in the quality of the data that's produced from the facility.
Something that still fascinates me is to see the embryo dividing in live and time-lapse imaging, to see how the maternal and paternal DNA gets together in the middle and form the one-cell embryo, and it's fascinating.
Recently, we've added a light sheet microscope to our core, and it enables us to image entire volumes of large tissue. This light sheet microscope is kind of revolutionizing how people perform sample preparation and then also acquisition and data analysis. We're able to acquire samples in their full 3D volume, enabling researchers to see intact tissues in ways that they haven't before. Another piece of equipment is our Andor Dragonfly system, which is what we call a multimodal imaging system. It's basically a Swiss Army knife. It can do various different techniques from just one microscope. It also is a turf microscope, meaning that we can look at a layer of fluorescence just above the cover slip. It's very good at cell membrane dynamics. It's also a super resolution microscope that has the capability for D-storm.
And that's why I got super excited when we got the Dragonfly and the light sheet, because for the first time we were able to track, live imaging for days, and the fertilized egg divides and that thing that before we didn't have any access, and we can answer questions about how fast they're moving or how slow they're moving, that if we're taking specific time points, we really don't know what's happening.
In addition to teaching people how to acquire images, one of the things we spend a lot of time doing is helping people with image analysis. So we use a lot of the ImageJ and Fiji software here. We also use Metamorph and Amaris. For someone to become a user of our facility, it's quite easy. You could email anyone in our group. We actually have an email address called microscopy@niehs.nih.gov. That email address goes to all of the staff members here.
Microscopy in general plays a tremendous role in our research because we use it to visualize the structures that we study, as well as individual proteins and molecules within that brain structure.
If I come to work one day and the facility disappear, I would cry. The problem is that we're not gonna have the tools to answer those questions, right? And yeah, I would be devastated, I think.